So the next person in the queue who will tell us is Vibhor.
Okay, so so you might have heard about my lecture on mapping single cell profile to large atlases of cell, reference cell. See, the vision which I made this was to actually to help clinicians to find precise cure, right? A precision medicine. So, like I told you, that if you could match a cancer patient cell to existing reference cancer cell from other patient who was treated with a medicine, right? Maybe we could give the same medicine to the other patient. That was the whole vision. Similarly, I mean, there might be some unknown disease which, if we can match it to certain other person who was cured with. So my vision is not yet there, because I think it will take one more decade to profile, like to have a large atlas of diseased single cell models with history of drug response. So it's a very long, it's a 2-3 decade project, but if you see, effect of drug has a context of cell types, right? Every cell type will respond differently to different drug, because every cell type, suppose, and currently for most of the drugs we don't even know the partners. We know one or two target pathways. We don't know other pathways. If you increase the doses, it may actually start hitting other pathways. Like metformin, if you see, we know mTOR and certain pathways. But if you increase the doses, which other pathway it will hit first? Right? It may become toxic, right? So, and drugs tried on animal model may not suit human. So now FDA has removed this criteria of testing the drug on animals. It's clearly has found now. That's why people are trying on organoid models, right? So, or humanized mouse model, but drugs being tried on animal, I mean, it's not necessarily that it will work on humans. So, okay, and then accordingly, the drug could have suitability according to age and pre-existing condition. So, if a young person is diabetic and if an old person is diabetic, maybe they need, of course, metformin is one that is global. But if you need to add something more with metformin, what else you could add for young person? What else you could add for old person? I mean, they may respond differently, and we have found that in our analysis, and then. But if we just try to solve this using matching single cell profiles, it might be tough, because I don't think it will take like next 2 decades to produce enough single cell profiles from diseased patients in every pre-existing morbidity and genetic predisposition, right? So what we need is to use currently existing knowledge and do some kind of systems level modeling, right? That is needed right now. So it can't be just solved using search engines and just like that, right? So we made an attempt. Actually, we published in Briefings in Bioinformatics 2 years ago. So what we did is that, I mean, so basically we developed I published a method in nucleic acid research where we could annotate, represent every single cell using the pathway activity. So for every single cell, we could transform the single cell transcriptome to single cell pathway scores. So for everything, so then we try to find how the pathways are co-occurring. You can say Crosstalk is a broader word, like how they co-occur or anti-correlated with each other. And then we took the disease gene set, and we saw which disease this set is correlated with which pathway, and we found that it has cell type specific context, right? So it is according to cell type specificity. So one disease may be correlated with one pathway, one disease gene set in one cell type, but not in other cell type, right? So we actually even normalized this correlation using 500,000 single cell profiles from human landscape data to make it more specific. And we did it for mouse also, same thing. What we found is that using for diabetes mellitus, the pathways which are associated with diabetes mellitus in mouse were different from in human.
Vibhor, let's keep to challenges.
Yes, okay. Yeah. Similarly for old, as we did Bayesian network modeling, right? We saw a difference in age. Okay, similarly, for cancer, so that was for non-cancer diseases. For cancer cells, I mean, Niranjan, who is not here, he was presented yesterday. He has a very good paper on using heterogeneity in clone-specific therapeutic vulnerability to, find drugs. So, and recently there is a paper which claim that they can also find combination of drugs according to the single cell profiles. But as we know that even if two cancer cells may have same transcriptome profile, their underlying epigenome might be different, right? They have their plasticity level might be higher or lower. Some may be more stem-like, some may be less stem-like. And the microenvironment of the cancer cell may influence, like if there are programmed immune cell around the cancer cell, they may exert different force, right? So we need many other dimensions to actually say that, yeah, in this patient, this drug worked, and the next patient, other patient, this drug will not work. Just answering this simple question. For this patient, we have this single cell or spatial single cell transcriptome profile, drug work. And if you have the spatial single cell transcriptome or single cell transcriptome, can we say with confidence that the same drug will work or not? Because even in the same patient, you have intratumor heterogeneity, right? So can we answer this question? So yeah, so even in spatial transcriptome profile, I mean, we can see different microenvironment, and you can have different epigenome, memory, poisoning. Cell have many other features, right, besides expression profile. And the main challenge is that even drugs are not specific, right? So for every individual, you may need different doses. So where are we? And this is my problem. So, yeah.
Thank you. Yeah, so Vibhor was talking about transcriptome. I will say transcriptome even doesn't match with the proteome. So, so the problem is not just within transcriptome. Even people who are doing proteomics does their profile match with the metabolome? So you can see the complexity here, and I think the dimension is quite large.